We're going to be considering this bottle to be one of your healthy cells. It has this plasma membrane, which is this plastic part, as well as it has your own genetic material, your own good DNA. It's about 3 billion base pairs long. And this is a coronavirus. And so what happens when one of your cells is infected by the coronavirus is the coronavirus has these spike proteins on its surface, and there's this receptor binding domain. And what happens is that these spike proteins are going to interact with something referred to as an ACE2 receptor, which is embedded inside of the plasma membrane of one of your healthy cells. And this is just like your skin, kind of. And so what happens is that when this interaction occurs, when the coronavirus infects one of your cells, it's able to inject its genetic payload, its RNA, into your cell. And this is now considered an infected cell. So in addition to your own good DNA, you now have this coronavirus RNA, and the purpose of this coronavirus RNA is to basically figure out how can it make a bunch of copies of itself inside of your cell and make the proteins that it's going to need to make in order to be a fully functional virus that can have a bunch of progeny and go infect even more cells. And so what happens if you've been infected? When we swab your cheek or we swab your nose, is that we'll be taking cells that contain not only your own good DNA, the DNA you had before, but also the RNA from the coronavirus. And so what we do once we've swabbed your cheek cells is we will put this into something referred to as a lysis buffer. And lysis buffer will break up the plasma membrane. It basically makes this whole plastic permeable so that we can get inside and see what kind of genetic material is inside of your cells, because we want to make sure that you don't actually have the coronavirus RNA present, because if you do, then we know that you're infected. And so basically what happens is once we put all this stuff in lysis buffer, the whole thing kind of starts falling apart. So like the genetic material that's inside is able to make its way out, and you've still got like plasma membranes and proteins and whatever other stuff is inside of your cell. But the two things that we really care about here are your own DNA and the genetic material from the coronavirus, its RNA. And so what happens here in the lysis buffer is that we will now centrifuge things. And because these things have physical differences in their masses, because proteins are a lot more massive and heavy and dense than the kind of genetic material that you have and the kind of genetic material that's in the coronavirus, as we spin this down inside of a solution, the really heavy stuff, like all these proteins and the plasma membrane and the organelles, are going to get pulled down really hard to the bottom of the centrifuge tube, and then the genetic material is going to stay up in the supernatant. The supernatant is the top liquid layer of the centrifuge tube after you've spun it down at like 500 Gs for a few minutes. These are incredible G forces that would totally rip you up, but the point is here that we are now separating physically the genetic material from all the other stuff in the cells. And so the issue now that we have is that we only have a little bit of genetic material to work with, because each one of your cells, it only has a little bit of DNA, and for us to actually be doing any kind of meaningful clinical experiments, we need to have a lot more genetic material to work with. And so what we're going to do next is something referred to as PCR. And so in PCR, what we do, and we're first going to make sure to do a reverse transcription on this actual viral RNA to make its DNA, so we have good genetic material to play with. So we're basically preserving all of the genetic material that the virus had. Then we're going to perform something referred to as PCR. The goal of PCR is going to be to basically make a bunch of copies of all the genetic material that's present inside of your cell. And so if you had the viral genetic material, we have primers that will help us make tons of copies of the viral genetic material. And then the next thing that we're going to be doing is pipetting this onto something referred to as a gel, as a page gel. And so what's going to happen here is that we're going to put all of this genetic material, which is including your own good genes as well as the virus's genes, into a well inside of a gel, and it's very much like Jell-O. And we're going to apply a voltage. There's going to be a negative side and a positive side. Genetic material inherently has a negative charge due to the sugar phosphate backbones that make it up. So the virus's genome as well as your own genes have this negative charge, and so we've got a negative side right here and a positive side right here. And so what's going to happen is as soon as I turn on this voltage, these genetic materials are going to get pulled through it. But because they're different sizes, because you've got 3 billion base pairs and the coronavirus genome has a lot fewer base pairs than that, they're going to get pulled through at different rates because one of them is a lot lighter. It's a lot smaller than the other one is. And so what's going to happen is that the coronavirus genome is going to be getting pulled much faster, and then we're going to turn off that voltage, and then we're going to look and see if we do see any genes or bands present at a particular location on this gel. So we already know a priori that if we applied some kind of voltage for a certain amount of time, we expect to see your genes as well as the potential coronavirus genes, if you've been infected, show up at a very specific region, and we call these bands. And so if we see the band for the coronavirus, that means that you do have the coronavirus genes inside of your cells that we swabbed from your cheek as well as your nose. And so if this is the case, then this is how we're able to confirm if people have been infected by the coronavirus. This test generally takes a few hours to complete because we've got to do a lot of steps, including, extracting these cells, doing the lysis buffer, amplifying the genetic material, doing reverse transcription to get the virus's genetic material in a format that we can work with. And so after a few hours in lab, we are able to figure out if you do have a band that tells us on a gel if you do actually have the coronavirus genes present. So if you do, then we're able to make the appropriate calls in order to quarantine people, as well as figure out what steps to move forward with in terms of treatments. So that is going to wrap things up for this video. I hope it helps. Let me know if you guys have any questions. Please stay safe and watch your hands, and take care.